Hello everyone, this is Mrs. Travai and these are the PowerPoints for science research. These PowerPoints have been provided by the Waxman Student Scholars Program at Rutgers University. Okay, let's get started. Today we're going to be talking about DSAP, the DNA Sequence Analysis Program, and how we start analyzing sequences. These next few presentations that you're going to watch are going to help you analyze your practice clones. So let's get started. Okay, so when we send out our samples for sequencing, we receive a file from Rutgers in this format. This is actually a waveform file shown in a program called 4Peaks. That's only a Mac-based program. We use a program called Finch, and you'll be looking at these in school in Finch. So let's take a look at what this actually means and how we analyze it. Just a review of where we are in our analysis. Right now we're determining, once we get our forward sequence, we used to have forward and reverse, but now we're just working with forward sequences. When we get our forward sequences, if they are of good quality, then we move on and we crop the ends. We're cropping out the regions of the vector. Remember that when we sequence, we don't sequence just the gene of interest, we actually sequence outside of that region in the vector. So now we know if the sequence data is of not good quality, we still have to submit that data. Rutgers needs to know that we analyzed sequences and we received data that was not useful or that was not readable. So the first thing you need to do is connect to the WSSP website, and the link in DSAP is found on their homepage. If you click on Current Project, you'll be able to find DSAP. And once you do that, you should have registered already, but if you haven't, you're going to register. And once you register, make sure you're selecting, if you're in the course, that you register as the class, and if you're in the club, you register as a club. If you're in both, you're going to register as the class, unless I've told you otherwise. Once we log in and you log in, you're going to see a page like this, and it's going to list practice clones. These PCs stand for practice clones. And it gives you a list of the clones that you're working on. When you first begin, you're just going to see the ones that are provided to you as practice. Okay, so a couple things on this page that you need to understand is that, again, the clone name is here, but actions here, if this is like green or it's highlighted, it could mean that there's a message for you. If you're analyzing the sequence and you want to continue, you click on continue. If you want to add a new clone, let's say you finish all 3 practice clones and now you're ready to add your unknown, then you could go here and add a new clone. Now, our clone table, again, the notepad is for taking notes on the clone. It's not used very much, but the clone discussion is the way that Dr. Rashan or the other staff members at Rutgers will contact you or communicate with you. So you want to make sure you use that link when communicating about a specific clone. If it's a general question, email me or Dr. Rashan with the question, but please talk to me before you start emailing the university. And the status of the clone just indicates at what stage you are at. So there are different types of status, and in this list here you can see there are many different types. The ones that you will see for practice clones only are reviewed and correct. Otherwise, if you're working on unknown clones, it's either submitted to NCBI, unreadable, no overlap, or unable to submit the sequence, or needs to be sequenced and reversed is another message that you might get. But that may show up as being worked on by a student. Those clones that need to be sequenced and reversed never really get closed out. So we'll talk about that in class. Okay, and that's the remark at the end is something that you enter in the program as you're analyzing the clone, and you will tell us. It's an easy way for the university to see which samples are coding or uncoding or not, or unreadable. Okay. And finally, at the bottom, the contact information is a general non-clone specific message, so to be able to contact the university. And that is the email that's provided at the bottom, wssp@waxman.rutgers.edu. This way, everyone who works on the program at Rutgers will receive your message and someone will answer it. Okay, so where do I get information if I'm stuck? There are some help. There's a help center listed in the DSAP program, and this allows you to click on when you're in a specific region. Click on help videos or help pages that have information. And you also have this help link, and it looks like a little life preserver to kind of help you out when you're stuck. And the other link here is the discussion link, which allows you to tap into that clone discussion message that goes to Dr. Rashan. Again, when you're first working on practice clones, you're going to need to start now. And initially, you may have to load the Mac program or the Windows program. They are free. You do have to fill out some information, but please just fill out the information that's required. They really don't contact you for any reason. And just make sure to deselect to receive messages so that you don't get any messages from these sites. But go ahead and do that and download those programs because they're what we're going to need to use, especially Finch. Okay, so the first step is to analyze sequence quality and to determine if the forward sequence is readable. So we're going to actually work on example sequence 1 to look at this. And here's the sequence in Four Peaks and in Finch. And we're going to take a look, and what we're looking for is distinct peaks. And don't be so worried about the ends in this region. Ends indicate that the machine couldn't really read what was there, but that's at the beginning of the read, and so usually you have ends at the beginning. Now, after you get past the 10th or 15th base, you shouldn't see those ends. So is this sequence of good quality? Remember, we're looking for distinct peaks, single peaks, and no overlaps. So what do you think? Yeah, basically this sequence looks really messy. A lot of this green in the background here, it looks like it's running behind a lot of the sequence. And then you see that these peaks, there's a red peak and a green and a blue peak. But you can tell that the sequence isn't of good quality by looking at how unevenly spaced these bases are. So if you see that, that's an indication. Are these good? No, right? Too many ends in the top, too many overlaps, and then the bottom sequence here, this green that's running behind it just seems like it was stuck there. And that may happen if the machine is not cleaned or there's something gummed up in the machine. So once you start examining the sequence in the beginning, if it looks good, you're going to keep following it until you follow the sequence out, and hopefully you find a poly A tail. If you find the poly A tail, you can stop looking at it at that point, because at this point, this is the end of our gene, and if it gets messy beyond there, we're not really concerned. So again, it'll get messier and it goes down here, it continues, and you see how messy it gets at the end, but we did get a poly A tail. That indicates the end of our sequence. So you select yes or no, and if it's no, you click. And you're going to get these questions to analyze. And it's going to ask you if this is a PC. After watching the video on bad sequences, answer below why you think the sequence was poor. If it's an unknown clone, go to images of your PCR and RD gel and determine what could have been wrong. There are many reasons why sequences can run poorly, and we're going to talk about that. And what experiments could you do to fix this problem? And again, there's a video online that'll help you figure this out, and we'll talk about that as well. So common causes for a sequencing problem. Here are some things that could go wrong, but the first couple are not very typical. So wrong or bad primer is usually not a problem. We provide Rutgers with the sample. They send it out, and chances are if they had a bad primer or the wrong primer, the entire run of sequences would be bad, and they would realize that. Dead sequencing chemistry, which means the reagents are poor, and they're paying to get these sequences done. So they should not be, the chemicals should not be bad. Blocked capillary, again, this is the company they're sending the samples out, and that should not be happening either. So it leaves us with number 4, which is more likely what's going to happen to your DNA sample, or what could happen to your DNA sample. 1, you could have poor quality DNA. It's just not a good sample, low yield, or mixed templates. So let's take a look at what that looks like. So poor quality template DNA is often caused by ethanol, and this is usually a problem in the mini prep. So students forget to do the second spin after the wash step. And when we get into the mini prep, you'll understand that this is incredibly important, or not doing the wash step, so that the SDS, the detergent that's in there, is not eluted with the DNA, or it's eluted with the DNA, so that it interrupts the sequencing program. It can't read the sequencing program. The machine can't work with samples that have those chemicals in them. So it'll give you no read, which is usually an indication of ethanol, more likely. The second one is low yield, and this I've talked about in the lab, and we'll stress over and over again that your steps in the mini prep are extremely important, and especially the first step when we take the sample and we put it in solution 1 and store it. If you don't have a large pellet to work with, if you didn't grow the bacteria enough, if the colony was too weak, then you're not going to have enough DNA to extract out of in the mini prep. And what you're going to see in the restriction digest is these faint bands. We always compare the cut and the uncut to the ladder. And in this case, the uncut looks okay. It's not as bright as this one, and it's probably as bright as the ladder. So deceivingly, you might think, oh, well, it should be okay because that's a sample that's going to be sent. And it may or may not work. So if it's the problem of low yield, then you get a read like this, where maybe it reads fine until you're out further, and then it starts to get ugly. So low yield can also lead to a high background or very small peaks. Now, what happens if it's a mixed template? If it's a mixed template, usually you can't tell in the beginning of the sequence, because all the sequences, every single sample that's sent out, has the vector in it. And the vector should be identical. So if you have more than one colony, if you picked more than one colony, or you contaminated your samples during the procedures, where you have more than one DNA sequence, or more than one plasmid, when we sequence the DNA, it will begin the same, so you will not see any problems. But right where the run of G's ends, right in this section, remember this is the run of G's that I talk about when we're looking for the start of the sequence. Once that ends, then you have a problem. You start seeing that there's something underneath the sequence. There's another band or another peak, and you have these conflicting peaks where it might give you a read, but there's another sample embedded in it. And the way to tell that it's a mixed template, again, is that the vector is identical. It's clean in the vector. It looks fine. But then you could see it's contaminated. Another sequencing problem that could happen is a quick stop of good sequence. Okay, so you have a good sequence. It's reading, it's reading, it's reading. It looks great. And then something happens. And it's an incredibly short sequence. And it could be that it's the poly A tail is right there. And it just it did not read into that region. That is a possibility. This could also, sequences after long runs of repeats may also give bad sequences. So for example, if you have this type of sequence where in the beginning it looked really clean, and all of a sudden you hit a repetitive T section. And usually in our bands, we've been seeing C's and T's where they repeat back and forth. And then after that, the sequence becomes poor. This, again, we can't use. So having repetitive sequences tends to do that. So what happens if you have a repeat section or a repetitive sequence, and why do you get all that overlap in this, after you read it? Because when we sequence the DNA, remember that it's not just sequencing one strand. You're taking many strands of the same DNA, denaturing them, and rebuilding them with bases that fluoresce, that will shine and will give you a read at different wavelengths. So these machines are able to do this, but polymerase, when it gets into a repeat section, may accidentally either omit a C. Okay, so this is the correct sequence read. This is an extra T is added. And then over here, we're omitting a T. So when we get to this point where it should read the same base, there are three different possibilities because one is ahead, one is behind, and one is correct. So you see this bump ahead, the correct, bump behind. So how do we know which is the correct sequence? You can try to clean it up and take a look at it, but at this point, if it's very messy, we're going to say it's unreadable. So again, here's another example. Before the repetitive section, very good. After repetitive section, you have a lot of overlap.